I'll probably wait for another 5 minutes so that more people can join. So maybe in the meanwhile, if there were any doubts from the previous week's session, previous week's lecture notes, I did get a couple of mail from people. People have mailed, which are in this week's session. So I'll touch upon some of those topics in this week's session. Um, we can go over if there are more doubts in this week's session. But if there are any questions from previous week's session, if there are any issues in accessing the videos or the lecture documents, you can let me know. So earlier the link like it was set in such a way that you have to ask for access, but I have corrected that now. So if you just open the Excel sheet that's linked along with the NPTEL course, you will open the Excel sheet and you'll have the links for the YouTube video as well as for the presentation slides that I'm using. So you can access them directly from there. You don't have to request access. So for people who were asking last class how to access the notes that I'm using, you can access it from the Excel sheet, and that Excel sheet is embedded in the NPTEL uh, website itself, the course page itself. If there are any questions related to how to access it or something, please let me know. I can recognize some of the names from the previous class, but I think there are also many new people. It's 6:03, maybe another 2 more minutes until I can start sharing the screen. But yeah, no one has any questions. Is everyone able to access the videos and the link to the lecture notes? Is someone who has attended the previous lecture, can someone just acknowledge that if you're able to access the videos and the uh, presentations?
Yeah, it was nice, Sanjeev.
Thank you. Thank you, sir. Thank you so much. Okay. I don't think any more people are joining, or like I'm not sure if people are gonna do what they did in the last class as well. Like I couldn't figure out any other way how I can like not keep doing this admit. But anyway, maybe I'll start sharing the screen, and if people do join later, I can stop that. Mm, share the window. Yeah. Is the screen visible in full screen mode? Could someone please acknowledge?
Yes, it is.
Yeah. Okay.
Yes,
Thank you
it
so
is.
much. Thank you so much. Thank you all again for joining for session two. As you can see, I'm sitting inside a lab. I'm actually in Bikaner now. I'm not at Bangalore at ISC. I have told in my previous session that I work on snake venoms. So we are doing some research here with snake venoms on camels. So I'm here in National Research Center for Camels in Bikaner, Rajasthan. So I'm actually in the field, actually in between work. So I thought like just this 1 hour session I'll quickly complete and get back to work. I'm sorry for all the background sound of all the equipment running and all of that. I'm really sorry, but yeah. So let me just get started. Okay, so the first week session was more or less like a general introduction to environmental biotechnology itself, what the field focuses on, what are all the techniques that are there, how we can use environmental biotechnology and the tools of environmental biotechnology to make things better for the environment. But in that previous class itself, we saw that superbugs, which are microbes, are very useful component of or a tool in environmental biotechnology, and we saw that we'll see more about microbes and the life cycle of microbes and ecology of microbes itself. And I also got a couple of questions from people in mail about the doubts that they have for microbial ecology and some of those topics. So maybe I'll try covering those, and if you still have doubts or if something is not clear, you can stop me in between. You can unmute and talk, or you can uh, put it on the chat box, and I can see that in the end or in after the class as well. You can mail me. So, microbial ecology, just like the word says, it's about the ecology of the microbes. Ecology is basically a study which deals with understanding the life history, the interactions, everything about a particular organism, all through the life cycle of that organism comes under ecology. It's basically everything that makes the organism uh, behave the way it behaves, or do some particular work the way it does. And microbial ecology is about the ecology of microbes. It deals with understanding the diversity of the microbes, and like as you can see in this image, there are different types of microbes, bacteria, fungi. All of these come under microbial organisms, and you know, we live in a world where soil has a lot of microbes, air, water, everything surrounding us, everything in the environment has a lot of microbes. And it's also a study that deals with studying not just the diversity, that is the different kinds of microbes, but also the abundance of each of them, how much is present in the soil, how much of it is present in the air, and so on. And the importance, importance could be about understanding their positive importance or negative importance. Microbes do cause negative effects to humans, so understanding their role in the ecosystem is also a part of studying microbial ecology. Microbes, and why specifically microbes are so important, not just here in environmental biotechnology, but in general, any biotechnological application, is because they live in a wide breadth of conditions. They can live in hydrothermal vents, which are extremely high temperature regions, saline conditions. They can live in cold conditions, so they have heat resistance, cold tolerance, salinity tolerance, and so on. Salinity is basically salt, so they can live in high salt environment. Generally, most other organisms that do not have this resistance cannot live in heat or cold because of it will, if it is too hot, it will melt them. If it is too cold, they'll freeze to death. And if it is too saline, that is, if the salt is also not in the right proportion, something called the osmotic pressure, which will change. Osmosis basically where the liquids move from a region of high salt concentration to a lower salt concentration. So osmosis happens in our body as well all the time. There are salts in our body which keeps moving in and out of cells, and the osmotic pressure is what maintains this flow of liquid between these different high and low concentration regions. So if the salinity is too much, if the salt concentration is too much, this osmotic balance will get disrupted and cells will just burst and die. So that's why microbes are special. They have these modifications in their cellular mechanisms which help them deal with these extreme habitat conditions. And microbial interaction and their relationships. So they just don't live in these extreme conditions, but they also interact with wide variety of other organisms between themselves. Viruses interact with bacteria. Archaea is also a microbe which lives in extreme conditions, specifically like hydrothermal vents and so on. Fungi, protists, and insects and nematodes. Microbes have this complex interaction with each of these systems. Microbes interact with plants also, animals also. So they are in the ecosystem in this kind of a very complex relationship. uh, Pointer, yes, yeah. So what are all the different kind of interactions the microbes undergo? So maybe this we can stop and go through in a little detail. The species diversity, that is, since I said that they can live in this wide range of temperature and salinity conditions, they are also very diverse, and this diversity is what actually facilitates a wide variety of positive and negative interactions. So these are some of the positive and negative interactions. So maybe this one by one we can go through. So here in this table, this column shows what kind of an effect the two interacting components will have. So this kind of interaction does not necessarily have to happen between microbes alone. This happens between microbes and higher order organisms, and between higher order organisms as well. But let's just understand the principle of these different terms and these different interactions. So this green color is species A and blue is species B. This is what we are going to look at in all of these cases when these two interact. But there can also be multiple interacting partners. This kind of a relationship can be there between multiple organisms. It can be a combination of these different ecological interactions, and they can form one community together as well. Those kind of examples are also seen usually in the ecosystems. But here for simplicity we will just look at two interacting partners and the ecological interaction between them. So the first one is mutualism or synergism. Okay, people are still joining. uh, okay. Yes. So mutualism is an interaction where the both the interacting partners benefit from each other. So here there's species A and species B. Both of them exchange components or interact with each other in such a way both of them benefit from it. The common example for a mutualistic interaction is the nitrifying bacteria and plants. So plants can't directly absorb nitrogen from the soil, nitrates and nitrites that are present in the soil. Usually bacteria decompose them. So where do these nitrates come from? We saw in the previous class that microbes are the ones that generally complete the food chain and the nutrient cycle and the energy cycle, because when some organism dies and it decays, it's the microbes that decompose them and uh, basically split apart all of the nutrients, carbons, oxygen, and everything, and convert them into different metabolites that are easy for the plants to absorb. So nitrifying bacteria usually, or bacteria that can convert the soil nitrates and nitrites into a form that can be absorbed by the plants through their roots. So these bacteria are generally found on the nodules of the roots of plants, usually leguminous plants. have this kind of bacteria, they support this kind of a mutualism very well. So this is also the reason why in India there's this practice called crop cycling, where they'll first put rice, and then for after harvesting rice they'll put leguminous plants, and that will help in fixing the nitrogen in the bacteria. So once the rice grows, it uses up all the soil nutrients, and now for the soil to again get enriched with enough nutrients, they'll now put nitrate, I mean the legumes, which with the help of these nitrogen fixing bacteria replenish the soil. So that's a kind of a mutualistic interaction. So what here happens is, yeah, yeah, what happens here is syntropy is usually where metabolites from one organism is used by the other organism for its own growth. So how does mutualism work? Let's take the example of the nitrifying bacteria itself. So the bacteria breaks down the nitrogen compounds in the soil, and thereby helps the plant to absorb the nutrients from the soil. And in turn, the plants fix the sunlight, they make food, the food that is prepared by the leaves with chlorophyll and everything. This is all like that we have learned in lower classes. The food that is prepared by the leaves is again then transported all throughout the plant. So roots also receive some of the food, and the nitrifying bacteria that live on these roots will utilize that food that is made by the plants for their own growth, and that helps in sustaining the bacterial colony. So this is what a synergism or mutualism is, where the components or the metabolites produced by one of the interacting partner is used by the other, and both of them benefit from each other. So here this graph, all these squares with different colors or metabolites, and each of these are the species, the interacting partners. So if you see this kind of an arrow with something like an end, this is to indicate that it is an inhibition. So inhibitions are usually there for regulations. We'll look that in detail in cases where it actually makes more sense. So the next form of interaction is predation or parasitism. Predation is seen in every organism, in especially in higher order organisms. A tiger feeding on a deer is a form of predation, and anything that eats or forages on something else is a form of predation. So even humans are also predators for some of the organisms that we feed on. So every kind of an interaction where one organism gains some kind of an beneficial effect, and the other organism gains a negative effect, is predation or parasitism. But yeah, before that I skipped commensalism, so I'll go to commensalism and then go back to predation again. So commensalism is a form of interaction where one organism neither benefits nor gets any harm, and the other organism benefits. So here, as you see in the effect, you can see that yeah, so the zero indicates that there's no positive or negative effect, and the plus is for indicating the positive effect. Pointer, yes. Yeah, so here the green organism is not gaining anything positive or negative, and the blue color organism is gaining a positive interaction. So this is a common example that we often see is ticks or mites that live on the body of cattle. Usually, they don't really cause much of an harm to the cattle. They're really big in size. They don't really mind the tick or mite being there, and the mite or the tick will suck their blood and feed on their blood and stay on them, and they also use them as a vehicle to move around. So this is a kind of interaction where uh, one partner does not have any harm or anything, and another partner benefits from that. So this example is a commensalism, and this kind of an interaction is a continuous food chain, because one metabolite gets transferred across, and the energy also gets transferred across. And the other one that I was explaining was predation or parasitism. Predation is a case where in both of the cases, one partner gains a positive effect and the other gains a negative effect. Predation is where a tiger catches a deer and feeds on it. Parasitism is where one organism lives on another organism and slowly starts killing it. These are the examples are usually parasitoid wasps, which lay eggs on other organisms, including cockroaches. They lay their eggs, and the eggs start developing from inside the body of the cockroaches and almost kills the cockroach. So here, the parasitoid wasp is gaining a positive effect, whereas the cockroach is gaining a negative effect. And this is not like a predation interaction. Predation is like usually when they feed on them and use them for their growth, but parasitism is a different kind of a positive and a negative interaction. And here, it's this is also food chain. The energy get transferred from one metabolite to another, from one species to another, but usually there is something that inhibits the growth or inhibits the other species. So there, the products from one species will inhibit the other species in this kind of an interaction. So this is food chain along with waste product inhibition. Another example is amensalism. So the difference between amensalism and commensalism is there is at least one interacting partner which neither gains any positive effect or negative effect. It does not have any effect at all. But in case of amensalism, the other interacting partner, which does not have any effect, the other interacting partner undergoes a negative effect. So this is the amensalism interaction. A common example again for this is in the case of penicillin, which is a product produced by fungus, is harmful to bacteria. But by producing penicillin, or by killing the bacteria, the fungi doesn't really get any benefit, but it is negative for the bacteria. So that kind of an interaction is amensalism. And this is not exactly a food chain because the fungi and the bacteria are not involved in a food chain, but some waste product of the bacteria is inhibited by one of the organisms. So this is just a waste product inhibition metabolic interaction. Another one is competition. Competition is common in all living organisms. It's there in microbes, higher order organisms. Even humans compete with each other for resources. So this is just a basic competition for resources. Here it's a substrate competition where both the interacting partners are competing for essentially the same thing, and they inhibit each other or they cause a negative effect on each other by competing for the same resource. So that's a competition interaction. And then the last one is no interaction at all. So there could be n number of microbes that coexist with each other without having any kind of positive or negative interaction with each other. So those are coexisting microbes that do not interact at all, and they also don't have any common shared metabolites that react with each other. So this is from this paper. So this is the reference for this chart, so you can get more information about different kinds of interaction and the metabolite protection in this case is from this paper as well. I'll maybe pause for a minute to just ask if there are any other questions that people would like to ask before I go further. Okay, I'm gonna take that as a no, but I'm still audible, right? Can just someone acknowledge?
Yes, you are audible.
Yeah, okay. Thank you so much. Cool. So the next thing is, yeah, this is where a lot of people have actually asked doubts about, I suppose. I suppose they're also attending this session. It's about species richness versus abundance. So what is the difference between the two? But before actually defining richness and abundance, let's just understand what a species is. So Species is just an arbitrary unit for classification of organisms. Species does not as such exist in the nature. Nature does not classify these things as such. Species is a unit that humans have introduced so that we can make a sense out of things around us. Several species concepts have been proposed about how you define something as a species. It depends completely on what you use to classify, or what mechanism you're using to classify a species, and whether it belongs to that particular species or not. The common species concept that are usually taught in schools and colleges, and something that people generally take into account is biological species concept, where when two partners interact and if they are able to produce a viable offspring, then they are known to belong to the same species. So if they produce, if they combine and produce an organism that cannot sustain the future generations, and if that offspring is not viable, then they may not be from the same species, is how the classification goes. So a common thing that happens in in laboratory conditions, like they make a cross between tigers and lions. Someone has any question? Okay, I think that was by mistake. Okay, so yeah, that was saying. So tigers and lions, the offsprings that they produce can be produced in a laboratory or in a zoo, but those offsprings are not really viable in the wild. They don't really, you don't see those organisms in the wild. So that's why they are two different species, and they cannot sustain a future generation by natural circumstances. Microbes reproduce asexually and do not fall under the biological species concept. So the concept that I just explained to you, the biological species concept, where two organisms come together and if they produce a successful offspring, then they are called as belonging to the same species. But what about microbes that do not need to have a male and a female gamete combining to produce viable offspring? So they just reproduce asexually by vegetative propagation and by spores and so on, and they also produce viable offsprings. So they are an example of something that do not come under the biological species concept. So how do we find different species of microbes? How can we classify them if we cannot classify them biologically? We can classify them phylogenetically. That is, phylogenetically as in depending on the DNA content of the organisms. Each of each of the organisms have their own unique genetic makeup. Sorry, yeah, the genetic makeup is very different for each of the organisms, and that can be used for classifying the organisms depending on how similar or different the genetic makeup of an organism is. If they have very similar genes, or if they have, say, very similar sequences for a particular gene, then we can classify them as belonging to a particular species. So that's how mostly microbial species are classified, based on the percentage of hybridization in their genomic DNA. Hybridization is a concept where when two different or two organisms come together, in any case, they mix and then they form a particular genetic makeup. Let's say there are two different microbial species, and they constantly exchange DNA in by some forms. So if microbes are not sexually reproducing, then how are they exchanging genetic material? They exchange genetic material by a method called horizontal gene transfer, where they can just transfer pockets of DNA contained in plasmids and so on to another organism. So this kind of exchange can happen between two different microbes, and if they keep doing this, their genes will keep getting combined, and this process is known as hybridization. Yeah. So this process is known as hybridization, and the more and more hybridization happens, there will not be any distinction between two microbial species. They will then be called as one and the same. So this percentage of hybridization can be a way to measure microbial species diversity. So coming to species richness and abundance. So now that we know how species in microbes are defined, we can take any particular ecosystem or an environment. Let's say, let's collect soil from one particular place, and we want to know what are all the different species that are present in that particular region. So just seeing the different kinds of microbes that are present, the diversity, measuring the diversity of the microbes or the different kinds of microbes or strains of microbes is known as species richness. When we are counting this diversity, we are basically just trying to understand how rich that soil or air or whichever that environment is. But in the complete contrast, species abundance is something, an indicator for the density of each of the species in this environment. So let's say there is microbe A and microbe B, and you have now taken and qualitatively assessed that there is microbe A, B, C, D, E, F, G. If you count the number of microbe A, the number of microbe B, number of microbe C in that particular environment, that would indicate the species abundance. So this is the basic difference between species richness and abundance. And increased abundance of a single species could result in an imbalance of resources. Usually, diversity of species in a particular environment is considered to be a good environment, and usually, an undisturbed environment would generally have high species richness or diversity, and not just abundance of one particular or one single species. And this will cause imbalance of resources because, let's say one particular microbe feeds on a particular substrate of carbon, it will keep consuming that and it will keep depleting that particular substrate itself. So for a overall balance of different substrates being consumed, species richness is important along with species abundance. But what also happens is, in cases of disturbed habitats, both the richness and abundance will start going down. If the richness is there and abundance is not there, okay, I'll just quickly change my network. I'm not sure if I'm still connected. Prasad, is your network better?